You have pore sizes that are larger, pore sizes that are smaller than that. And as you accumulate more particles, obviously your pore size is getting smaller. But if you would use something like this is I don't know what kind of filter it is. I cannot remember. Yeah, probably. I'm not sure. But doesn't matter. This is a beautiful coccolithophore, no? This is a scanning electron microscopy image. But if you look at the stuff behind, that's actually a filter. And you can see these little dots. These are the pore sizes. This is actually 0.2. So, I mean, there's a big difference between this and that. This is a little bit more exact. I mean, there's errors, of course. But it's a little bit more exact. But we just always use the GFF just because, all our methods are developed with GFF. Historically, everything is GFF. But always remember that this is 0.2, and molecular people are using that, and this is 0.7-ish. So we are losing some stuff here in between.
This is highlighted circle. Go back.
I have no idea. I think I snatched this from probably a grain of something. This is from a paper by Greet Nukermans. She did quite a lot of, I think I'm going to talk some of her stuff. She did lots of SPM work, suspended particulate material work. And I think in one of the papers where she's explaining the correct methodology, how to do suspended particulate matter, I think she actually went and took photos, images of her filter as a function of something. I don't remember. But I can pull out the paper because I would still like today to show you how to properly measure both carbon and SPM. So Emmanuel was talking a lot about, there was now the whole discussion here about the assumptions that you make once you're deriving things from optical measurements. Because, optics is cool and quite a lot of people are interested in optics themselves, but I'm one of those people that I'm just using optics as a tool to study something else, And you saw it, guys, yourself, how long did it took you to filter that chlorophyll, prep, blah, 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 blah. And it takes lots of time. And chlorophyll is actually not the most anal one. That one is one of the easiest and fastest one. As you get to different techniques, it gets a little bit more and more harder and more expensive to collect these samples. So usually it turns out to be that parameters that you're interested in, that you really want to measure, so like chlorophyll or carbon and stuff like that, are time consuming. And so their sample resolution, there's so many parameters that sample resolution is small. But there's only so many parameters that we can measure with a high sampling resolution. So for example, if you really want to have a really good knowledge about distribution of the carbon, distribution of the chlorophyll, taking these samples every 2 days or 2 hours when you're in a cruise is not enough, But the thing that we can measure along the way, the small number of parameters, and these are usually physical parameters, including optics, that we can measure on a high scale. So once again, think about your ACS. It's 7 hertz. So 7 times a second, it gives you 82 wavelengths of A and 82 wavelengths of C. It took you, in order to do the chlorophyll, you had to go collect the water, you have to filter the water, you had to put the filter in the acetone, wait 48 hours, and then process it. Long time, lots of time and lots of mula, no? So usually, again, it turns out that these biogeochemical parameters are the one of interest, but they're, we cannot sample them often enough. The physical parameters, they're all over the place and they're super easy to sample. So what we start doing is developing these relationships between the biogeochemical parameters, the parameters of interest, and these physical parameters, And in a sense, artificially increase the sampling rate of these parameters of interest, And this relationship is something that we call proxy. You can call it an algorithm, but when you're talking about in situ optics, usually you talk about proxies. When you're talking about remote sensing algorithms that allow you to use radiances to explain, to measure biogeochemistry, you talk about algorithms. But pretty much they're the same thing. They're kind of like a substitute, a fake way to measure things of interest on a proper scales without spending lots of time, And that's kind of what I do a lot, because I'm really interested in biogeochemistry. I'm interested in phytoplankton economy. Phytoplankton economy, I'm interested in phytoplankton ecology. And I'm really interested to understand the way that this phytoplankton community structure modifies the flow of carbon in the ocean, and then kind of how does the physics play a role in it. So I'm going to be talking to you today a little bit about carbon and not about geochemical proxies, that we can develop using all these tools that we kind of like talked about this week. And this is just a random selection of the papers along the way. It has some meaning in my head. Hopefully it's going to have some meaning in your head. And I was really trying to pick the papers of the previous people who took this class previously, just so you can see that, you know, there's hope for you. I'm going to focus on ocean. I kind of pulled in a slide from freshwater. I know there's like, you know, you're doing freshwater and maybe somebody else, you're doing kind of like, you know, complex estuarine systems. But the story goes everywhere the same, you know, like, you know, climate change, everything's bad, things are bad. If you put stuff in a model, it looks even worse, but the uncertainties are gigantic. And uncertainties are gigantic just because the parameterization of these climate models is bad, because we don't have really good knowledge of every single little thing that happens to this carbon as it's traveling through the atmosphere, and especially when it's entering water bodies, regardless if it's ocean, even more or less for rivers and freshwater systems, no? But if you think about it, what's happening in the ocean, you have this carbon entering, you know, just dissolving. You have happy phytoplankton, as I told you before, takes in carbon, you know, gives us oxygen. This carbon now changes its form in some sugars, and there's lots of little things that are going to go try to eat that sugar because it's a source of food, no? So you can have these different types of phytoplankton getting eaten up by small zooplankton or larger zooplankton. This phytoplankton can also die and start making these aggregates that we call marine snow. You have aggregation. And so what happens with this carbon that is now taken up by phytoplankton, it can aggregate and sink out. Portion of it is going to get respired, and portion of it is going to get into the zooplankton that is ultimately going to poop it out, you know? So this is pretty much, you know, lots of things happening with this carbon. But if you think about it, all the things that we said that you can measure, you can use quite a lot of these optical measurements, optical tools to measure these things. For example, we know that we can measure phytoplankton, no? We can differentiate different types of phytoplankton. We can measure aggregates, no? We can measure the change in the phytoplankton. We can measure the change in number of aggregates. We can measure the sinking of the aggregates. We can measure lots of different things. So optics is really good to get a good idea of what's happening in the ocean. And because these proxies allow us to, you know, collect much, much, much more data, we can start talking about these things on a proper scale. So these, once again, these optical tools give us a higher resolution data set. And once again, traditional methods are time and money consuming. And using optics, using all these optical proxies allows you to sample the parameters on a scale of importance. So let's say you have a bloom and you're really interested in, you know, understanding the whole bloom, but you have a ship and you went to sample your chlorophyll, and now there's a really rough weather, you can't go out there, and you're going to go there again, and bloom is gone, no? But if you had some kind of autonomous vehicle or mooring that's measuring this chlorophyll and stuff like that, you can actually figure out stuff, what's happening with the bloom. You don't have just 2 data points. You have the whole story. Because if you have just the beginning and the end of the story, you can start making really weird hypotheses. I always use this example. I used to have it was not my cat. It was my boyfriend's cat. It was never my cat. So the boyfriend's cat would get food every morning. He eats a little bit, meow, 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 goes away. Suddenly the food starts disappearing. You know, I come in, you know, I feed the cat, come back from work, food is gone. It never happens. And it keeps on going. So I start making hypotheses because I have 2 data points. I feed the cat, the bowl is full, I come back, the bowl is empty. You know, and the cat is the same like, you know, thin, skinny, crazy cat. So hypothesis 1 is like, it was an inside-outside cat, so the kitties, you know, from the neighborhood are coming out and partying on the food. Second thing, cat has an internal parasite, no, and it's probably going to die. And what turns out to be, I found a possum living behind my washing machine. I was living in LA. And what this possum was doing was eating this food. Since I didn't have a camera, I just had 2 data points. I couldn't make, I just guessed hypothesis. I didn't have a continuous observation to actually allow me to understand what's happening, what process is happening that's causing my food to disappear. So optics allows us that, to watch continuously these biogeochemical processes so we can make good hypotheses and understand what's happening in the ocean and take that knowledge and put it into the models and ultimately in climate models, okay? Other good stuff is that these optical tools, since they're giving us lots of data, you could use it as a validation for remote sensing and for validation for existing models. So you can do 2 things. You can build the models and you can validate the models. You can build the proxies and validate different proxies. So they're really, really important. So many people, you're going to see these graphs. I really like them. I think I have another one. where you can see the processes in the ocean of the interest in different time scales and different spatial scales, no? So I study phytoplankton blooms and zooplankton grazing, so everything what I'm doing, it's happening in order of a day to a month, and somewhere from 10 meters to 10 kilometers, no? So if I don't have good measurement capabilities and measurement techniques, I'm gonna miss my bloom. So you can find many of these graphs. This one is a different color, so it's like, they're like, ooh, they figured it out, but it's pretty much the same thing, no? It talks about a little bit different processes, but allows you to understand the scales of your process and how do you have to plan your sampling in order to actually capture what's happening inside of it. So another reason why these proxies are so awesome is that technology has evolved, and we have these smaller and much more robust optical instruments. You guys played yesterday with the VSF and with BB3, but that BB9. Well, BB9 is, you can minimize it in teeny tiny puck and put in a bunch of these instruments. This is a drawing of the wire walker that Melanie over there is doing her PhD thesis on. And it has like instrument package up here. This is like maybe the instrument package is oscillating really up and down faster the water column with this teeny tiny puck on it. This is a manual. This is autonomous vehicle 1.0 carrying stuff. I just had to put him here because I love this. A glider, mooring, not a glider. This is a sail drone. What's the name? Tara, Giorgio with float. So there's a bunch of these little platforms on which we can put these instruments and continuously measure optics and try to derive biogeochemistry for them. Once again, you're not dependent on rough weather. The coolest things in the ocean, for example, from my perspective, are happening in North Atlantic in the end of the winter. I'm never going to be able to get there because nobody's sane in their mind will go there. No captain is going to take you. But if you have autonomous vehicles, you can really actually start studying the processes they're leading towards the spring bloom. How do you do that? Well, this is the citation from Colin from 2015 class. Anything that causes variability in a sample is an opportunity to extract additional information from that sample. So we were teaching you about the fact that, there's a certain trend that you want to follow, and then there's that scatter around it. It might be uncertainty, but once you correct for the uncertainty, the rest of it might be a really cool story. That's why we're all the time, understand your measurements, understand them, but then the stuff that you have on the side might be really leading you to something cool. And I'll show you a couple examples of that. So when we're developing proxies, again, Emmanuel, what's the name? Inert?
Extensive and intensive.
Yeah, you wrote it down. Okay, intensive and extensive. Okay. So you can try to develop proxies for quantification of the properties of some kind of bulk thing. So you can do, of course, chlorophyll. We did chlorophyll. You can use chlorophyll fluorescence. You can use absorption to explain the chlorophyll. You can do particular organic carbon. It has a really nice relationship with the scattering. As you heard for the last couple of days, things scatter. Things in the ocean have carbon. Therefore, carbon scatters, and you can make that relationship. You can look at the suspended particulate matter, so amount of sediment, amount of total suspended particulate matter, and that scatters again, so you can do backscattering in CP. You can actually measure the particulate and organic carbon. In the ocean, that is usually, from our perspective, kind of hidden in those coccolithophores, And coccolithophores, except for scattering like crazy, they polarize the light. So what you can do, you can take your C-star, your attenuation meter, put a polarizer, and one with and one without, and actually start differentiating between total carbon, total suspended particulate matter, and just PIC, so particulate and organic carbon one. There's actually quite a lot of publications by Jim Bishop, who's using that to quantify the carbon export in the ocean and accounting for particulate and organic carbon as well. Dissolved organic carbon, you'll see that sometimes measuring CDOM works to explain it, sometimes it doesn't, most it doesn't. Actually, you can start measuring get some ideas about the distribution of the nutrients, the yum-yums that phytoplankton needs to grow by using the UV absorption. There's instruments to do that. And then you can start going into realms of primary productivity. But that actually should be on the next slide, which primary productivity is actually telling you something about the change, about the flux, about the capability of phytoplankton to convert that carbon into the sugars. And then you can go to these other ones that are extensive variables where you're going to start looking at the ratios. And you can start talking about the particular composition. Emmanuel said, again, backscattering ratio allows you some differentiation between the mineral particles and organic particles. You can do backscattering ratio like BBP over BP, but you can also sometimes, especially in the ocean where the absorption is low, you can use CP and look at the behavior of that one. You can look at the particle size. Emmanuel talked about it a lot. It gives you an idea what sizes are you encountering in water, but really you have to be careful about that. You can use the slopes and the fluctuation that Emmanuel mentioned. I actually found that Nathan actually put it in a paper. I'm a co-author on that paper, so it is published, so it doesn't matter. I'm the second co-author. I wasn't paying attention. I was fighting productivity. It doesn't matter. You can look at phytoplankton composition. You can use many different things. You can use the fact that their chlorophyll to carbon ratio is different for different types, so you can play with that. And you can look at the type of the dissolved organic carbon. So these are all the things that are associated by geochemistry. And then you can start looking at the fluxes, the changes of the rate. So you can look the increase of the total carbon. So if you measure carbon continuously, that increase is going to tell you something about how is that carbon being produced. You can look at the change of the particular organic carbon into dissolved organic carbon. So talk about the fluxes between two pools of carbon. You can look at what's happening with the carbon in the vertical column. How is the carbon getting exported through aggregates, through the poops, or through something else? You can look at the productivity, so change of the carbon pools over time, and also that type. I said that. I know this is a lot, but I'm going to go now into examples. It's going to become a little bit more. So once again, this is kind of what we're going to focus on. I'm going to try to go through different parts of this schematic to kind of tell you what's happening here. So chlorophyll biomass, I'm not going to go on that a lot. Colin talked about it and everything. But this is one of those examples when you plot things on log scale and plot things on a linear scale. If you plot things on log scale, it looks much better than a linear scale, no? So what Colin did here with her co-authors, I think Manuel included and a bunch of other people, she took chlorophyll measured from HPLC from a bunch of different cruises, a bunch of different experiments. So there's that approach, you minimize the bias due to the people who are measuring stuff. And try to compare it with the chlorophyll coming from the chlorophyll fluorometer, because we have chlorophyllorometer, and if you ask any physicist, that's your chlorophyll, no? When you start looking at that chlorophyll, there's problems with it, no? And she was trying to see, is there a simple way to translate a chlorophyll fluorometer into the HPLC? So if she plots it if she plotted it on a log scale, it doesn't look that bad. But once you start plotting it on a linear scale, you start seeing, scatter. You start seeing different distributions. And then it don't really seem to have too much with, I know you didn't, this is like with the parts of the ocean here. You see some kind of behavior in light blue, no? There's a behavior in blue, there's a behavior in pink. There's some structure to it, no? So we can start looking what is the reason for it. So in that same paper, what she did is she used slow factors. So look at the calibration, difference of calibration between the one that comes from the factory that is bad and the one that she did in the lab, and showed that there is a variability associated with the type of algae she's using. So I don't know if you guys remember, it was a big lecture when I was talking about the phytoplankton, but I was telling you the type of pigmentation, we can use HPLC pigments to differentiate different types of algae just based on their evolutionary perspective, because they evolved their pigments in a similar way. So these are those lineages. And so she separated the calibration, differences in calibration based on these lineages. So you can have this variability in chlorophyll fluorescence to chlorophyll ratio just based on the species composition and origin. When you look at the chlorophyll fluorescence to chlorophyll ratio again, you can start seeing something like this. So what is plotted on the y-axis, chlorophyll fluorescence to chlorophyll ratio in both cases. And this is the photosynthetic available radiation. So that's PAR, the amount of light. So what happens in the ocean, and Colin, I think I was out of the room. Did you talk to them about MPQ?
Nope, that's coming next week.
Okay, so Colin's going to talk more about it, but there's something that we call quenching, no? So you know that the little chloroplasts or chlorophylls are living in this protein complex, and they're their magic, and they're like, oh, awesome, light, yeah. And then it starts being a little bit too much light, no? And what happens when there's start being a little bit too much light, you have things start turning off. The chlorophyll starts getting damaged. And the way that the energy is, once it's absorbed, is distributed in a cell, it can go into the heat energy. It can go into the, I'm going to need too much into detail. It can go in a production of photosynthesis, or it can go into fluorescence. What happens here, they're just getting completely freaked out by the light. And you can see that in that fluorescence measurement. So you can't always use chlorophyllescence as chlorophyll, because in a high light, it starts dropping down. If you have different species, it acts differently, okay? So it's really not a good measurement. Here, this is from a think my paper, this is from the Jing et al. Paper, and you can always see the same behavior. When it starts reaching the really high light, your chlorophyll fluorescence to chlorophyll ratio is going to start dropping down. Because the chlorophyll fluorescence is not a really good measurement for chlorophyll.
Have you, or has anyone actually ever taken a look at like how the chlorophyll concentration changes with the intensity of light at a certain location for time? Let's say you measure fluorescence for 48 hours at one specific location because like
Or in a,
there's clouds and
or in a lecture, or in a lab. You can do it in a lab. Yeah, yeah,
have you done that?
yeah. People have done it. Yeah, she's going to talk about it next week. So to think about, there's two things. There's chlorophyll concentration. There's the chlorophyll fluorescence. It's how much chlorophyll you have and what is chlorophyll doing. No, think about it. Those things. Take your plant, buy a basil plant, put it next to the window. It's going to start turning really light green, and after a week, take it all the way back in the darkest part of the room, and she's going to start becoming super intense green. What is happening? That the plant is starting to increase amount of chlorophyll inside to account for the change in light, no? And those kind of processes happen on a scale of a day or two. This response happens really fast, on a scale of hours or so. This is a physiological response to too much light. If you continue doing that, you can start damaging the cell. This is just like initial response that this behavior happens in the middle of the day, and as the day goes off, you're going to see somewhere around 6 p.m., this quenching is going to start disappearing, chlorophyll is going to start going back. So, for example, if you want to use chlorophyll for validation, you might want to either use the chlorophyll during the night or do a correction. And there's many corrections out there. I'm going to, of course, push stuff that I was working on. So this is your chlorophyll, again, versus chlorophyll fluorescence. So what we did, we took into account, we developed a nonlinear correction that take into account light, of course. We did depth, makes sense, deeper, less light, more light. Time, which actually is really not a function of anything, but in our case was the availability of the nutrients, and that will change your fluorescence. And we managed to correct it more or less. So we had some good relationship that was working for our crews. There's a global Zhang. You said it's pronounced it differently. I need help here. Zing or Xing? Xing. Okay. Xing has, in last, I think, 8 years, published some really good, well thought of papers about how to correct this chlorophyll fluorescence, because we have this chlorophyll fluorescence on floats, and we don't have chlorophyll samples on the floats. So I've been thinking a lot and trying to find different ways to correct that quenching and correct this chlorophyll fluorescence so we can actually use it for something. So this is just one of the examples of this correction where he used the light again, and then what's happening in the water column, the stratification capabilities of the water column. Is the water column well mixed or is it stratified? And if you're going to do something, use, Emmanuel's on this one, so if it doesn't work, blame him. But these are really well thought papers. So one other thing, all this paper, all this stuff, I'm going to give it to Sasha Doppler. If you click here, it's going to take you to the paper, okay? This is going to be a really good literature thing. Okay, so this is about chlorophyll. I'm not going to talk about that stuff because you're going to get a lot about it next week from Colleen. No, she's the chlorophyll person. I am more of a carbon person. So once again, particles scatter light. You guys heard that, no? And the ocean, open ocean, especially dominant particles are carbon-containing particles because we don't have, except for the sandstorm, sources of sediment, no? So the particles scatter. You can use attenuation or some kind of scattering measurement to measure the particles. If the particles have carbon, you can build a relationship between attenuation, scattering, or backscattering to derive your carbon. That's what you want to do. So the first person who did it, actual relationship with carbon, was Gardner, Wolf Gardner. And he sometimes floats over here to the class. He spends the summer in Maine. Maybe that's going to happen. But this is an original JGOVs. In 1993, he published this relationship between the carbon and attenuation meter. And you can see that it's more or less a linear relationship, no? And they worked really great because now, even in 1993, before any of you was born, he could actually, figure out the distribution of the carbon on the frequencies of his attenuation meter. That's awesome, no? That's really great. So many people since then took and started going, massaging it, and I was one of those people and started looking, what's happening, what's good, who does it best, or me or him. So I also did the same thing, and this is one of those examples that Emmanuel was telling you. You don't go and plan the experiment, but it kind of dump the data on you and you deal with it. This is my dumping the data on me and dealing with it. I took all the measurements of the carbon and plot them as a function of attenuation, and I start seeing some relationship. Very similar to Gardner et al. There's different lines of different relationship. You see some variability, okay? Not bad, no? And then when you start plotting backscattering versus carbon, my line was, which one is my line? My line is probably the red line, and all these other lines are other relationship. So it's going a little bit crazy, no? And these are different data sets that I'm comparing, but the relationship is a little bit crazier. Why do you think that's the case? Why is it a little bit more stable between carbon and attenuation versus carbon and backscattering? Yes, so Emmanuel just talked about a lot. So what attenuation is what? What is attenuation dependent on? No, you have a beam of light and it's hitting a ball. Cross-sectional area, no? But when it comes to backscattering, remember the VSF. It's kind of different for different types of particles. Then if it's an aggregate, it's kind of a little bit different. And then we did something else. And then when you have a different indices of refraction. So what I tend to call backscattering is a promiscuous measurement. And I don't think it's a good thing to say, but it depends on lots of things. It's not very simple as attenuation. Backscattering picks up lots of things. So if you try to use backscattering as a proxy for certain things, it's similar to chlorophyll. It's not really a clean thing. It's a little bit, picks up a little bit on everything. So what turns out to be, and I think I kicked out this slide, turns out to be that each of these relationships are highly dependent on the type of particles that these people encountered. So for example, this one here, where there was Balch, he saw a lower carbon per higher backscattering than me. So what you can see right away that he probably encountered some particles that scatter higher than my particles. He was sitting in the middle of Coccolitha for bloom. Remember chalk? Scatters like crazy, but for the same amount of carbon that I've seen. On the other side, Stromsky, he went down to Arctic, and he was encountering really large, gooey cells. I think he had actually, what's the name of the thing? I'll remember. He had a large gooey cells. They were not really optically so intense as my cells, so he was getting more carbon per backscattering. So here, the differences were just based on the type of the cell, the internal structure, morphology, indices of refraction. So once again, backscattering, it's very, very, promiscuous measurement. So what I did next is okay, I didn't saw any variability, but I saw some variability. So I started looking at those things that Emmanuel was talking about is mass-specific attenuation. So I just flipped it over here, and this is the carbon versus CP. So I was looking at the slope, but that's in a sense upside down mass-specific, carbon-specific attenuation. And what I've seen that there's some kind of function, there's a relationship between that mass-specific carbon-specific attenuation and the maximum amount of attenuation. So I started feeling, okay, why is that happening? Well, it seems that as you're increasing attenuation in the open ocean, this is open ocean, you're entering in a range of blooms. And the open ocean, who's blooming? Phytoplankton, big phytoplankton, diatoms, no? So you're actually increasing, you're going from smaller to larger cells, no? But one important thing that happens here is that you're going from small cells to larger cells. Who remembers that I was telling you something about diatoms? What is a specific thing for diatoms? They're glass boxes, and inside of them they have a balloon. You remember? I was telling that something. So the structure of the diatom cell is that you have a box, and inside of the box is a balloon. So everything that is yummy in the cell is pushed all the way to the edges. But if you look at other cells, dinoflagellum and stuff like that, they don't have that balloon. So when it comes to looking at the cross-sectional area versus the amount of carbon in the cell, diatoms actually have a little bit of carbon in comparison to their cross-sectional area. But dinoflagellum, if they look at them, they look like a potato. I always call it a potato because it's so dense. It has so much DNA and so much carbon. So for the same cross-sectional area, it's going to have much more carbon. So here, as we are transitioning into the areas of diatoms, we're getting less carbon per cross-sectional area or per attenuation. But as we're going here in the areas where there's no blooms, where we have potatoes, we're getting much more carbon per attenuation. There's just like a little relationship, but it seems to be holding. So that's a proxy, no? That's a proxy, that's that proxy with E, what I cannot remember. Where is that leading us? So I was telling you, this is just, I put it in, so I wasn't planning to think about this, but we were start talking about biases, you know? And how do the biases affect the data? So this is our current POC algorithm. This is a NASA standard POC algorithm. It was developed by Stromsky in 2008. And what he did, he took a relationship between particular organic carbon and blue to green ratio, no? And blue to green ratio seems to work for anything what you want to measure in the ocean because, yeah, of course, it's associated with chlorophyll, so why not? Chlorophyll-containing particles have carbon, and it's okay. It seems to be working. That's what we've been using since 2008. But we start looking at the methods, and since I haven't shown you yet how to measure carbon, I'll show you tomorrow. When you're filtering for carbon, you have to be so anally clean, no? And one thing that happens is that dissolved organic carbon start messing up your measurement because that dissolved organic carbon that is present in the ocean starts attacking the filter, and you have to account for that. And if you don't account for that, you can actually make big errors in your measurement. If you don't filter enough, then that amount of carbon that is dissolved, that is sitting on your filter, is going to bring large noise to your signal. So what he did, like, well, it's okay, I'm just going to filter lots of water, and my signal to noise ratio is going to be good, so I'm not going to count for the amount of carbon that is absorbed on that filter. People from my lab did a modeling exercise, actually empirical, and they developed a model to understand how much of the carbon gets adhered to that model, and it seems that's a lot. So I was like, okay, so since I'm in charge of these algorithms in the house, how much is our error in our current algorithm? Because this is a bias, but it's a bias depending on the amount of water you filter. So I don't have his volumes. He never reported the volumes. Went back to the paper. He said he filtered 1 to 8 liters, blah, 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 blah. So kind of like, you know, this is an exploration. I pulled this out of my ass pretty much, no? But I followed his paper, and I said, okay, if this was me, for this amount of carbon, I would filter 1 liter. That's enough, no? For this amount of carbon, I would filter the maximum volume that he says. And somewhere in the middle, I would filter 2 liters. I put that correction, and what I'm getting is I'm switching the amount of carbon from blue dots to red dots. And I'm refitting his algorithm. So now his original algorithm was this purple line, and the new algorithm, they corrected for bias. How correct that is doesn't matter, but I'm making an assumption. They corrected for bias is the red line, no? And I'm going and recalculating the global carbon. How much does it affect that? Well, so upper panel is showing you a distribution of the global carbon for 2018. And this is around 50% of the global carbon. So you can see here, this is the amount of carbon from the satellite. Just look at the blue lines. The majority of the carbon in the ocean, the majority of concentration of carbon in the ocean is around 40 micrograms per liter. I told you that the error can be 30 micrograms per liter. If you actually take the bias and propagate it, it seems that we've been overestimating majority of the carbon for around 30%. So, bias that was not accounted for is currently causing us to overestimate global carbon for 30%, which is a lot, okay? He didn't, you know, the uncertainties were not defined. We went back to get them by ourselves, made assumptions about his stuff. So we now know that there's a big bias in our data, and we have to go and develop a new algorithm. So this is a bias. It's really hard to propagate. It's really hard to account for. But it makes a terrible mess, okay? Phytoplankton carbon. So you have a total carbon, that's your POC, particular organic carbon, but it's not only phytoplankton total. I mean, it's not only phytoplankton there, you have bacteria, you have detritus, and so on, but really want to know phytoplankton carbon, no? Because that's the one that is making all the magic happen, no? So a bunch of people have been measuring number of phytoplankton, let's say using IFCB, the one that I was showing you yesterday, and then you calculate the volume, and from the biovolume you make some assumptions about how dense is the cell, and you can calculate carbon. Or you can use a much more analytical technique, which is pretty awesome, that Jason Graff is doing a lot, where he sorts the cell using a gigantic flow cytometer, cell sorter, and he is collecting a cell of a single type in little tubey tubes and actually measuring those cells. So he's actually measuring real analytical carbon and then dividing by the number of cells. So he actually has this carbon per cell. So using that, he actually calculated this analytical carbon of all the phytoplankton that he encountered and the backscattering associated with it. He got, you know, we can discuss this relation, but this is pretty good relationship, no? This is great because now we can measure carbon using backscattering, and we can measure phytoplankton carbon using backscattering. The problem is both of them are linear, and they don't behave linearly in the ocean, but it doesn't matter, no? So what can we do with this? Well, for example, we're currently now going to start instituting at NASA the phytoplankton carbon algorithm, so you can get carbon from space and you can get phytoplankton carbon from space. So we can, using something called GIOP that Jeremy's probably going to be talking about and Colin later on. It's a model that allows you to derive inherent optical properties from the radiances. We can calculate backscattering. And then we take that relationship that Graf has developed, and we can calculate phytoplankton carbon. And you can start doing cool things, no? So this is a plot that is showing you the anomaly of the phytoplankton carbon in a permanently stratified ocean since 1998 until today. And you can see going up and down, up and down, up and down, no? You can see some seasonality. So this would be like, you know, probably your southern hemisphere spring, northern hemisphere spring, southern hemisphere spring, and so on and so on. And it's always much more intense in southern hemisphere because it's predominantly ocean, no? And then you can plot the ENSO index. This is upside down ENSO index, which means this is La Nina, this is El Nino, and you can start seeing the trends. We did this before all the time with chlorophyll, and it was really not clear. But once we start doing it with the phytoplankton carbon, it's so much better. You can actually see the things that you're assuming to happen because chlorophyll, again, chlorophyll is great, but it's only chlorophyll. Carbon is a much better measurement of a biomass of the phytoplankton because it's not dependent so much on the nutrients and happiness and physiology and so on. So this is something that you can do thanks to that proxy that somebody developed, no? So you can do climate. I didn't do too much suspended particulate matter, but it's really important to measure when it comes to the coastal areas. You can connect it to the attenuation, you can connect it to the backscattering. And it's really, you know, when you do coastal areas, if you're doing geology or anything like that, I have something stuck to my butt. I mean, it's a really important measurement. So this is from the paper that Grit Nukermans published, and she's the same one with those filters that I was showing you before, where she did an amazing study of a relationship between suspended particulate matter and a bunch of particular properties, so like attenuation, VSF, so on. And in this paper, she developed a really good relationship between backscattering and CP versus suspended particulate matter. The reason why she has a really good relationship, once again, is because she's anal. Her analytical methods when it comes to the measurements of suspended particulate matter are amazing. So she can develop really strong relations. See, look at the uncertainties, everything is plotted here, statistics is amazing. So she developed these relationships that really hold across many areas. So while she was doing this, she developed a method that I'm going to show you today, which is a really good method to use to measure suspended particulate matter. But once again, things scatter, they can be carbon, they can be anything. You can use it to the derived amount of things that you're interested in. When it comes to dissolved organic carbon, this kind of gets a little bit problematic, no? Because we can really not measure dissolved organic carbon. We can measure colored dissolved organic matter, and not all dissolved organic matter has color, no? So when it comes to the coastal areas and riverine situations, especially here, I mean, here in the Mariscota estuary, you very often get a nice relationship between the dissolved organic carbon and absorption of CDOM on certain wavelengths. So if you're working in these areas where you have a stable source, one or two sources of a CDOM, and that source is replenished and you don't have photodegradation and processes like that, you can actually build a nice relationship, and you can use your ACDOM to track the plumes of the rivers. No, but when you come in the open ocean where the sources of, first you have less dissolved organic matter, you have much less colored dissolved organic matter because there's so much light and there's not a continuous production like in coastal areas, that relationship starts falling apart, no? However, a recent graduate from this class has recently published an algorithm, a global DOC algorithm. I have not read the paper in details, but I'm obliged to tell you there's a global DOC algorithm, and he's claiming that you can use absorption derived from remote sensing to derive global dissolved organic carbon. So link is here, you guys should look at it. But just think about it, you know, like what is the probability of absorption of the sedum to be closely related but dissolved organic carbon in the open ocean? Go ahead.
In the Arctic, it actually works well because the
Yes.
Arctic has so much fresh water.
Exactly.
You get a reasonable
Yeah.
sea out of it.
And I mean, and Colin is kind of like eyeing me under the glasses, and I'm sure she has something to say. This is, you know, she's a sedum queen. But in these freshwater environments, you can expect it to kind of hold. But then there's so many degraded processes that are degrading this color, this old organic material. They're really not related so much with the processes of degradation of DOC. It's not comparable. It's not always a good thing. Probably this is going to work in low salinity waters, but once you start getting mixing and different processes, things happen. Aggregates form, light shines. Different types of salinity are going to start changing the chemistry of the usually colored material that this relationship is going to start falling apart, and this is what you're going to start getting. Colin, please.
I would just mention that, you know, if you look at material for example, it's really difficult to come up with a good relationship to
Yeah.
DOC. It's just because the composition of the
Exactly.
material being washed into the rivers is in so many different areas.
There's so many different sources.
So many different aging processes. There's so many
Yeah.
different microbial processes. And so you just, you know, it's just, even looking at sort of the rivers going all the way around Florida, from the all the way down to the Blue Current, there's been some really nice work
Yeah.
done there. And it's just really difficult to get these relationships. So they work locally.
Yeah, they work locally, and you were saying that. So, like, you know, probably if you work in something like the Mariscota River,
Yeah.
you're going to be able to develop your little local relationship that you can just keep on going. But don't ever think that you can use that relationship somewhere elsewhere. Yes, try to use the technique, the idea, but don't take things globally. And that's why I was saying, read this paper and tell me what you think about it, because I'm not really sure that it works. But again, I haven't read it into details. The chaos. This is my favorite part. So that's that part where what Colin was saying, there's an opportunity, there are things that kind of fall off that one-to-one line that I was showing you up to now. And this is where, like, you know, where you have to start recognizing that opportunity and chaos and things that don't fit the lines as you expected them. And once you have your uncertainties sorted out, then you can start looking at the things that are sitting on the outside and start figuring out, is there a nice story here? There's something else that I can develop. And this is where it usually comes to the composition. You know, the scatter on one-to-one line is probably because you messed up something, but it's probably the impact of the composition of the particles, type of molecular composition of the dissolved organic carbon, so on. Those are the stuff that are going to pull things away on the side. And that's the chaos that I like a lot. So this is the graph that Emmanuel showed you yesterday. Oh, I thought that I changed this. Ooh, I think I was changing somewhere else. Probably I did. I'm going to show you something else then. This is that graph that Emmanuel showed yesterday where Nathan and company looked at the variance over mean in scattering to estimate the change in the size. No? So you can use that variance over mean to see something about the size of your particles. So in his PhD thesis, he did the same thing, but he used in situ data. This was happening during North Atlantic bloom. So first he did that using chlorophyll. So chlorophyll fluorescence, he did variance over mean ratio. And if idea is the same, what that should give you is the amount of chlorophyll fluorescence per particle. If your size of your particle is going up, you should have more chlorophyll. So he made that assumption and plotted the variance over mean ratio. And he's seeing that when the diatoms are blooming, you see the increase in that variance, increase in amplitude. So he's saying, okay, these are diatoms here, there's a small phytoplankton here. And he confirmed that using microscopy and other data. But then he did the same thing with backscanning. He wasn't really following the story. But what it turns out, that what he was actually seeing is process of aggregation. When phytoplankton bloom reached the peak, the cells kind of going senescence, and when diatoms go senescence, they get sticky. When they get sticky, they start bumping into each other. When they bump into each other, they make aggregates, and these aggregates sink. So what he's seeing here is a process of aggregation, no? Increase of aggregates in the upper part of the water column, and then the number of aggregates goes down, no? So that's what he managed to see with that variance over mean ratio. So that's that chaos that I'm talking about. He used the opportunity, some kind of crazy measurements, to say something about the quality, the composition of the particles that he's seeing. This is not really a chaotic approach because Meg is not chaotic, but this is a publication of Meg Estapa, who took the class with me, and she's the one that has the deepest ACS. So her ACS can go up to 5,000 meters, 6,000 meters, because she was really interested to look at the particle composition of the particles and what's happening with these particles that are coming out of the hydrothermal plume. So she went and deployed C-star, LIST, AC9 down inside of the plume, and she was looking at the PSD slope, the one that Emmanuel was talking about. Down here and down here, you can see the change in the particle's size distribution slope. And she was also looking at the ratio of LSS, which is a side scattering. It's not the best measurement, but if you don't have backscattering, you can use it, or CP. So that's something of a similar to backscattering ratio. And she saw some really nice features that she later on associated with the types of plumes that she was encountering. So here, see? So this is all happening in 5,000 meters. So you don't have to do optics only in the surface or a shallow bottom. You can take it all the way to the bottom of the ocean and start looking at the processes where you really don't think that you can use optics. Let me just, oh no, I actually did later. Okay, so then you can start looking at the phytoplankton community composition. I think Sasha is going to be talking about this a little bit more. Yes. So just to show you also, like a paper done by Ali Chase is going to join us whenever she shows up. And what she did is this amazing analysis where she took the absorption, did Gaussian decomposition, and tried to relate each of these little peaks to the pigments, to the pigment composition. And using absorption, she could actually retrieve tons of different pigments and look at their distribution as a function of the path, the Tara ocean stake. It's a really cool thing, you know. She started decomposing the absorption and you know, got so much more. She took this a step further and just recently published a paper where she took the same approach to remote sensing reflectance. She wasn't able to you know, derive this amount of pigments, but you know, the results were really good. So check that one out. This is the ACS data, no? So it's not the filter pad. So once again, think about 7 times a second, she can tell you something about the phytoplankton community composition, which is pretty cool, no? I mean, you average everything over 10 minutes because of the signal to noise ratio, but still every 10 minutes, which is, if your research vessel is going usually velocity of 8 miles, every 1 minute, sorry. If you average, so if your ship is going 8 knots, you're pretty much getting a data point every 200 meters. Am I doing this calculation correctly? Probably not, but yeah. You're getting very abundant measurements of the phytoplankton community composition. You know? And then you can go and be cheap like I am and not go and do fancy math, but just plot things versus things because I'm a biologist. No? So this is the second part of me, my postdoc, where things got dumped onto me. No? And so I didn't have too much to work with. I just had chlorophyll and backscattering. Okay, so if you think about the phytoplankton has chlorophyll, phytoplankton has carbon. No? We know the carbon is associated with the backscattering, but it's kind of like problematic, no? And then I have chlorophyll fluorescence, which is even more problematic because it's kind of suffering from light, it's suffering from community composition impact, it's suffering from nutrient availability impact. So I tried to still plot things versus things because that's what I do. And I found kind of 3 behavior, no? Behavior 1, behavior 2, behavior 3. And it kicked out all the data that was quenched, so I didn't look at that. So what I figured out that once I plotted the amount of carbon, the amount of diatom carbon in my total phytoplankton carbon, I started seeing somewhat of a relationship between that and chlorophyll fluorescence to backscattering ratio. So what this is saying, as the amount of diatoms are increasing, your chlorophyll to carbon ratio is increasing. And that's kind of following the theory because we know that diatoms usually have a higher chlorophyll to carbon ratio inside of the cell, no? This is cool. So okay, let's take this knowledge and go back. So what this is saying that this and this are no different, the same communities, no? And this is something completely else. So what I figured out later looking is that this was a teeny tiny cells, like a small teeny tiny phytoplankton. It wasn't diatoms, no. They're just a recycling community that came after diatom bloom. This was the happy diatoms, big large cells blooming. And this, they ran out of the silica. They ran out of silica, they start freaking out, they changed their chlorophorescence, and they completely changed the pattern. I did some stats, blah, 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 and I had that I could apply to data coming from 4 gliders and 1 float. So I can extend it, but just within the scales of my paper. And that's really important stuff. I didn't extend it past the point where I had the first measurement in time and last measurement in time. I just stayed within the scope. So if you're going to be using this, and I see people use it a lot, make sure you make this relationship for yourself. This works only in surface. Okay. Okay, so this is year day. Okay, so we come there, the spring bloom starts. Sorry, I wasn't explaining. They ran out of silica, freak out mode, and then everything disappears, and the little picoplankton shows up. What I'm showing you here is the data from the Lagrangian float, which was just following 1 water type. So I can actually see this as a function of the time within 1 water mass.
Did you get a percent diatom product?
So percent diatom carbon product is the percent of the diatom carbon within the total carbon. So I used flow cam data, similar to IFCB that I was showing you, and flow cytometry.
So did you have discrete samples?
Yeah. So all these dots are my discrete samples that I compared to chlorophyll to backscattering ratio. So this is what I was after. I was after the number of diatoms. I took that, compared it to my optical proxy, to my optics, and I developed an optical proxy. I took an optical proxy that tell me a story about what's happening, and I took it to all the autonomous vehicles. So from, there's around 40 data points, I went to 200,000 data points or more. Yeah? It's voltage. Doesn't matter. It's you can calibrate. It doesn't matter. It will never give you anything good. So I keep it in voltage. Voltage is just the output. No, you remember, guys, when your backscattering was outputting, you were writing either the counts or voltage, no? And then you took that and applied the calibration. So on. I just didn't apply the calibration because I don't think that makes sense. It usually doesn't. So I just kept it as a voltage because it has all the information in there. I didn't even try to correct it. And that's, if I tried to correct it, the plot that I showed you earlier that was corrected, if I tried to correct it for time, light, and everything, this would be suppressed. This would actually go straight. I would lose that chlorophyll increase. Go ahead.
You calculate the carbon from the images, carbon diatom, carbon products. Is it with
Biovolume?
biovolume?
Yeah.
BSD, or?
So this was done by Mike Siraki and Nicole Polton from Bagel. These are our collaborators. So you have different models. So you usually measure the size of your particle, and there's different methods. What they do, they have really good models to estimate different types of different shapes of phytoplankton. So from the image, they make a 3-dimensional one, fill it up, and then use probably Mendenhauer and Bessart. A certain relationship to switch from the biovolume to the final carbon would take an effect that these are diatoms or these are not diatoms. So pretty much you take your image that we collected yesterday, you try to make a 3-dimensional cell out of it, you calculate the biovolume, and then you do the conversion with the carbon. It's not the best way is actually to do analytically, as I was telling you before, the stuff that Jason Graff does, where you sort the cells and actually measure the carbon in cells. Here I'm again making assumptions. And eventually one of these days, Jason Graff and Nicole Polton are going to sit together and compare the data they took 5 years ago on the same cruise with the idea to actually do this comparison, because we still don't know for a fact how does the biovolume, the carbon that is collected through the biovolume and assumptions about the modeling is compared to the one that they're doing analytically. Yes, go ahead.
Where was this?
North Atlantic.
North Atlantic. And so the story is they're happy and they run out of silica
Mm-hmm.
because they use it all up.
Yeah.
And then they die and the tiny ones come.
Yeah, and then they die. Same data set.
Same data set.
Same day they die,
Aggregate.
they aggregate and they sink out. And I'll show you the rest of the story how they sink out as we're going. But this is all the great thing about this this experiment that everything was done through optics. We developed the whole story and everything through optics, all the way to, like, you know, Melanie's advisor, Melissa Mann, who was a graduate of this class, figured out a completely new way and confirmed something that people were theoretizing about, a new way of export of carbon to the deep ocean. And everything thanks to optics and autonomous vehicles. So, okay, so now I'm going into the composition of the carbon, and this is, again, this is the approach that was done in, you can see Chesapeake Bay in the rivers of the Atlantic midcoast. So what they looked at the slopes of the CDOM in 300 to 400 region, no? And they figured out that the slopes of the CDOM, absorption of the CDOM, and that, I have to finish up soon, I still have shit tons to go, that actually is relatable to the molecular mass of the dissolved organic carbon that they were encountering in the water, no? And that worked really good for them. And you're going to see that this paper has like 300 citations, and people are just using the same assumptions, which is once again not correct, because Colin said, and we're going to say, these relationships might hold in your one little river that has a specific type of carbon coming into it. But once you have a complex rivers where you have different types of carbons coming inside of it, this might really not hold well. So be careful. But I went and did it here in Maine. I was working with some crazy microbiologists, and they were like, we have this iron oxidizing bacteria. I don't know which carbon they're using. Help us, help us. I'm like, ah. So one did the same thing. I just collected some samples of the water. I can show like all these like little yucky, muddy ponds that look like poop and orange, reddish stuff inside. Those are the iron oxidizing bacteria, no? So just went and connected some water, and over the period of time, I was measuring the absorption of the CDOM, and I looked at that slope and the change of the slope. And didn't say anything about molecular mass. It just said the change of the spectral slope. And it seems that the community composition of these iron oxidizers, which I don't know anything about, is dependent on the amount of the dissolved organic carbon and the spectral slope, so availability of a different type of carbon and iron that is linked to it. So, you know, like, optics takes you in weird ways. But, you know, again, think about different ways that you can actually use these measurements to talk about different community composition, different processes. You know, crazy iron bacteria that are sitting down the road. I still have fluxes. You know what? I can go through it. Okay. And yes, I have Will Smith. Okay. So this is so you know, all this stuff that I was talking about stocks, composition of the stocks, and now we're going in the realm of the fluxes. So change in the stock. So either the carbon goes from one thing to another, or goes from one pool of carbon to another, or it starts sinking, or goes somewhere else. How to use optics to measure it. This is the best video ever. So this is collected on North Atlantic. So this is exports. Melanie is your video probably. This is a video of a camera. Melanie's advisor breaks cell phones. She breaks everything. And she had broke one cell phone, and she put it in a housing and put it underneath a sediment trap. On the bottom of the sediment trap, there's a gel that captures particles as they're falling down in the water column. So she starts collecting these videos of the things falling. So this is a deployment of the sediment trap over a period of a couple of days, and you can see these little particles, this marine snow, this carbon sinking into it. And it's pretty awesome. It doesn't have anything to do with optics, but, you know, it actually tells you what's happening. And then you're going to see zooplankton going inside, try to snatch stuff, and yeah, oh yeah. Wait. There's gonna be a little jelly.
Differentiate day and night.
Yeah, look at part. Look at the jellyfish. And it's gonna go like, yeah! Look at this. Ah! Awesome, great. No, magical things happen in deep ocean. How can we get to them? No. Well, first we have to figure out what's happening on the surface. How does this production of the carbon happen? Who's eating who? What amount of carbon goes into respiration? What amount of carbon goes in production? Blah, blah, blah. Can we use the optics for it? Of course we can. We can measure carbon, we can measure chlorophyll, we can measure light. All these things are important. So what Melissa Umand, graduate of the class, is doing, and she's hopefully going to give me something pretty soon, or not. And this is something that many people have done before. You can look at, in a constrained environment where you have a stable environment, not influx of different types of waters, you can look at the variability of your CP, which is actually carbon. And what this is telling you, in a period of 1 day, do you have a growth in the amount of carbon, and then you have a drop. And then you're going to have a growth, and you have a drop. What this tells you that there's a carbon being produced and then disappears during the night, probably during the respiration, and it goes up and it goes down. So when there's light, it goes up, then it goes down. So it tells you something about the primary production, it tells you something about the respiration. So you can tell lots of different productivity estimates from here, Other thing that you